So I want to thank, first off, the MagLab for inviting me to give a talk tonight on some of the research we've recently done through FSU and at the MagLab. I'm Dr. Pete Morton. I'm a visiting assistant research faculty in geochemistry at the MagLab. And tonight I'm going to give you a few details about a recent trip we took to the North Pole. And so tonight's talk is titled Mapping the Chemical Properties of Arctic Waters. And I'm going to lead with the slide right here, which doesn't look like much, but this is a very much magnified image of an Arctic diatom called Coscinodiscus. And you can see here that it's got some nice chloroplasts in here, very green. There's all different kinds of these guys that are living up in the Arctic, and so they'll play very well into the story, even though they're not chemistry per se. They are very much involved in the cycles that I'll be discussing. So a little bit about myself. I've been a research faculty at the MagLab, again in the geochemistry department, since 2015, so since I got the project to go to the Arctic. Before that, I was a postdoc with Dr. Bill Landing, who's here in the audience with us tonight. You may have heard him play sax anytime within the past few years. He's quite well known, and deservedly so. But yeah, so I joined Bill's group in 2009. Had a great time doing, looking at trace metal cycles in the ocean. I'll tell you a little more about trace metals in a minute. But before that, I got my PhD from Old Dominion University in chemical oceanography. And so this is what I do, and I hope that you guys enjoy what we're going to share with you. So last September, we made it to the North Pole. That was a very big deal, and we were very excited. There were 50 scientists, 90 Coast Guard crew on the icebreaker, the U.S. Coast Guard cutter Healy. This is a big research vessel. For those of you, just kind of give a refresher, Coast Guard is kind of like the policeman of the ocean, and but it's a great platform to launch research. And so this is a very unique and special ship, great crew, and lots of unique features that make it perfect for doing some good science in the Arctic. This is us actually at the North Pole, and we took a little picture here. You can't see, but we do have a pretend North Pole there just for kicks. But yeah, this is us. This is the group that made it up there. 2015 was the first time that anyone, at least from the US, made it to the North Pole unaccompanied. And so it was a very big deal, very well publicized. We didn't hear about it. We were in the middle of nowhere. But you guys may have heard about it. But any other time before then, we've usually had to have multiple ships kind of working together to make it to the North Pole. So this was definitely a historic event. This was on September 7th, 2015. So on this ship, the scientist party actually included 3 of us from FSU. So Dr. Bill Landing, who I mentioned before, shown here playing saxophone at the North Pole. All right, Bill, is it every continent now? Every continent. He's played sax, and that includes Antarctica, so no small feat. Plus the North Pole, not really a continent, but it's kind of hard to get to. Also, you'll see in this picture Dr. Neil Wyatt, who's also with us in the audience, back over there with Bill. He's Bill's postdoc now, and myself posing with St. Nick. We were on the good list. So what you'll notice here is that I'm also standing on the ice, but that is not the Healy in the background. That is actually the German icebreaker, Polarstern. And this, so not only was the September 7th occupation of the North Pole a historic event for the US, it was also historic in that we actually met at the same time with the German team who were doing complementary work to what we were doing. And I'll tell you more about how all that fits together. So we actually had 2 trace metal scientist parties occupying the North Pole at the same time on the same piece of ice. So it was a really interesting kind of side note there. So what's the big deal about trace metals? Why do we even care about them? They're trace. They're probably low concentration, not all that important. But in fact, they actually play a very major role in how things cycle through the ocean, especially when it comes to the biology. So the ocean is more than just salt and water. And a lot of times, I think it's better to think of it as a very dilute soup. It's packed with nutrients, got a lot of other things in there just besides salt and water. So this thin soup supports the growth of microscopic organisms, including marine plants called phytoplankton. And these require certain nutrients to grow. I've shown here a picture of another Arctic diatom. They're very different in structure and capabilities. And so it's just like a zoo up there. So in addition to the major nutrients, like nitrate and phosphate, the phytoplankton also require very low concentrations of micronutrients that we call trace metals. And these are thousands of times lower in concentration than nitrogen or phosphorus. And I think a good way to think about trace metals is think about your multivitamin, your one a day. So you're going to eat your food, your 3 balanced meals, but then a lot of times we'll supplement our diets with these one a day vitamins. If you look at the list of ingredients on the back of those multivitamins, a lot of those ingredients are the same things that we're looking for out in the ocean. We're talking about things like iron, vitamin B12, copper, zinc. These all play important roles in plant and microscopic plants that play important roles in our body as well. And so that's a really important thing. It helps control how these plants grow in the Arctic. So despite their low concentration, these trace metals do play key roles. And in the biogeochemical cycling of every major nutrient. So not only are they important, they help other nutrients cycle through the environment. And that includes the major ones, carbon, nitrogen, and phosphorus. What I'm showing here are some of the highlights. So we've got some carbon chemistry going on here. It's facilitated by zinc, cobalt, and cadmium. We've got phosphorus chemistry facilitated by zinc-driven enzymes. We've also got the nitrogen cycle, which is a really important cycle in the ocean. It's like a whole suite of metals play roles in different enzymes. Iron, molybdenum, nickel. There's a bunch of them. There's a whole bunch of different enzymes that all depend on these trace metals. The most important ones, I think, arguably, without hurting anyone's feelings, whose favorite metal may not be represented here, but iron, zinc, cadmium, and cobalt are probably the most important ones. Some of the things that why they're so important, besides the fact they have a biological role, is that they're actually very difficult to measure out in the environment, especially in the middle of the ocean. So they're at very low concentrations, which usually requires very large volumes of seawater, because you got to get enough seawater to concentrate it down to bring the metals to an appreciable level that you can actually detect in some sort of instrumentation. The matrix, the background that you're trying to look through is not a nice clean background. It's not a clean solution. It's very salty. It's very complex. So if you're talking about concentrations that are like parts per billion, parts per trillion, even lower, but the salinity, the major salts, like major ions like sodium and magnesium, calcium, these are at the percent level. So in addition, they're actually, these metals are very contamination prone. So the things that we're looking for out in the ocean, that's what most of the stuff we're traveling on is made out of. So this is not an active research vessel of any sort, so don't get the wrong impression here. But ships are big metal things that are always bleeding off metals. If you think about how, besides the fact that it's, a lot of these are made out of iron, a lot of the sacrificial anodes you'll find on ships are made out of zinc, and that's the purpose of those anodes is to bleed into the water. So if you're trying to look at zinc concentrations in the ocean and your whole ship is just like leaching out iron the whole time, it's a really difficult obstacle to overcome. So to study trace metals and do it right, it requires specialized methods, instruments, and really importantly, you have to have the right people. It takes a lot of training. You kind of have to develop a consciousness, I guess, to making these sorts of analyses. Because of these challenges, much of the trace metal chemistry of the ocean remains unknown. All right? So it turns out that FSU and the MagLab, this is the right place to do this kind of research. So I think I'll focus on the people here, but the MagLab, especially back over in geochemistry, our division that is actually out of the MagLab, we have a lot of great instrumentation and a lot of great laboratory space that is super purified air. So we have HEPA filters blowing positive pressure all the time, keeping the environment dust-free. We have to change our clothes, change our shoes, wear caps when we go back into these labs so that we don't get our dust into the samples. And it takes a lot of you can be the right person with the right kind of mindset, but if you don't have the right facilities, it's just going to be an episode in frustration. So, but again, coming back to this other thing, just because you have the right space doesn't mean that you've got the right people. And so one of the persons is Dr. Bill Landing again, who in 2003 with a couple other colleagues developed a very clean, very fast sampling system for collecting trace metal samples from the water. And the first time they deployed this was in the Atlantic back in 2003. And it had a team of about 4 to 6 trace metal scientists that did the operation. And these would be the people that actually deployed the equipment, took the sample bottles, brought them back into the laboratory, did all the subsampling, did a lot of the shipboard analysis. So 4, maybe 6 people did a lot of the work. All right, I want you to keep that number in your mind. So here's Bill here. He's usually the tallest guy in the picture. So this was actually sponsored. This is a risk they took, the CLIVAR program. And CLIVAR stands for Climate Variability. And these guys have been going out to sea for years. They're pros at it. They make a lot of great measurements. They do things like the temperature, the salinity, the pH. They look at the dissolved inorganic carbon like CO2, bicarbonates. What's the other one? The chlorofluorocarbons. They make a lot of these measurements that have to do directly with carbon cycling in the environment. So on this 2003 trip, they actually included a very small, again, 4 to 6 people, trace metal component to see if we could actually do this sort of work very quickly and very cleanly. Well, it turns out it worked, and worked very well. So fast forward a few years, in 2010, 2011, the first U.S. GeoTraces expedition studied the North Atlantic Ocean with a little bit larger sampling system. So this one had actually 24 sampling bottles instead of just 12. It could collect larger volumes. It could do more. It could measure more depths or sample more depths at the single time. And to operate and do all the sampling for this actually required something like 35 scientists. So we've definitely scaled up in operations. So things are changing very quickly in just a few short years. Things went from just like a small 4-person team to something like 35 people, scientists from all over the U.S., putting their heads together. And this is actually an international program, GeoTraces, and I'll tell you more about that. But yeah, so in 2010 and 2011, we actually took a trip from Lisbon, Portugal, down to the Cape Verde Islands, and then picked up the second half of the trip from what's called Bermuda and back down to the Cape Verde Islands. So we actually covered the whole east-west transect. Again, that was 35 people, a lot of people on a ship doing a lot of work. We're all specialists. But if we're ever going to try to cover the entire world's oceans, we're going to have to get everyone's help. And so what you'll see here is the result of that with a team up of many, many other nations. So let me see if I can get this spinning. So what you're looking at here is a map that is definitely a pretty cool thing. The U.S. contribution to this is this bit right here. Neil was actually on the UK trip, did something in the South Atlantic, similarly down here. And what you're looking at are the concentrations of dissolved iron in several transects across the Atlantic, north and south. And so you'll see the hot spots are these red. Those are really high concentrations. You usually find those over hydrothermal systems, mid-ocean ridges against the shelf. You can find some of this in the surface here, right up at the top. That's actually coming from Saharan dust that's falling into the ocean. A lot of dissolved iron coming from that. And all of these this map really kind of is just brand new uncharted territory. So again, this required teams from the US, Germany, the Netherlands, France. Other countries are getting involved in other basins as well. But it truly is an international effort. Everybody kind of has to work together. So the Arctic is a different challenge. We're not dealing with open ocean, tropical climates. You know, those are pretty nice cruises to go on. These are a little more challenging. It's really cold, and it's not just uncomfortable at times, but it's actually, it just totally wreaks havoc on equipment. You've got to deal with a whole other kind of set of logistics. You'd be surprised how quickly things will freeze. I was surprised, and I knew better. I knew that things were going to be cold. I was still just overwhelmed with just how quickly things would break. So what we're showing here is the map of the U.S. GeoTraces CLIVAR joint expedition to the Arctic. So in order to get more bang for the buck and also to make better use of our resources, this became a combined effort. So the original team that originally sponsored Bill back in 2003 has now joined forces with the latest iteration of that, that's GeoTraces. And so what we've got here is this really just amazing survey of the Arctic Ocean that gives us not only the chemical parameters like that are involved directly in carbon cycling, but also in that same context we have all of the trace metals, and as well as a bunch of other parameters. So this is a really full cruise. There were about 51 people on this cruise. I mean, it was a really big deal. And so what we did is we left out of Dutch Harbor, Alaska. If you guys have ever seen Deadliest Catch, that's where that show is filmed. That's where those guys base their operation. So we left out of Dutch Harbor. It came up to like this, the shelf here, worked through the Bering Strait, up along the western side to the North Pole, and then came back down on the eastern side and back home to Dutch Harbor. So what you'll see in these pictures is that it's not really open ocean here. You reach a point where it's completely covered with ice, thus the need for an icebreaker. So in summary, we had 18 full profile stations, meaning that we went from the surface down to the deep, all the way. So we got a full profile of everything happening from top to bottom. In addition, we had 7 marginal ice zone stations. So marginal ice zone stations are where there's not full coverage of ice, but you're starting to hit little pieces. And that can be anywhere from like 10% coverage up to 90% coverage. But basically what we're looking at are pieces of ice that aren't completely kind of sealed together. And then we did 6 full ice stations where the ice was so frozen over and thick enough, at least 1 meter to 2 meters thick, we could actually get out on the ship, walk around on the ice, do some research, and then climb back on the ship and then break away to another site. So again, we've gone from a 4-person team to a 35-person team to now a 51-person team, not including the 90 Coast Guard personnel required to keep the ship running as it is. So just to show you how this kind of fits together, this was a tag team effort because GeoTraces is international. This is a tag team effort with the Dutch and German, who were on the Polar Stern, and that was the blue research vessel I showed you in the other picture, as well as with the Canadians. So what I'm showing here is that we're looking now top down at the North Pole into the Arctic. So here's the US GeoTraces cruise right here. This is this red line. The German-Dutch team followed this black line right here, and this is where we crossed over right about here, a little bit south of the North Pole, but pretty close. And then the Canadians had this transect right here where they cut through their archipelago along the north. So why now? Why is it so important that this was the next research expedition? Certainly a lot easier to get to some other oceans. We've got some great data from the Atlantic, got some great data from the Pacific. The timing for going to the Arctic it was really critical. We know that changes are occurring. We know that the air is getting warmer above, and then actually we've seen very good evidence that the waters below the ice, below in the Arctic, in the deeper layers, are actually getting warmer as well. And as it turns out, it's those intermediate waters, those waters that flow in from the side, that actually tend to have a really big impact on the Arctic system. So what I want to show you is this is kind of like an animation of surface and deep currents and how they kind of form and move into the Arctic from the Atlantic Ocean. So we'll start, oops, sorry, let me go back one. Here we go. So I'll just kind of walk you through. So right here in Florida, this is where the Gulf Stream comes off, and it works its way north. It's nice and hot. It's nice and salty. And by the time it gets up to the north, it really starts to chill. So once you start cooling off, that salty water gets really dense and heavy and will actually sink down to the ocean floor. And so you've got the surface currents coming up from the south. They get up into the colder regions in the north. They'll sink down and they'll start to flow out along the bottom. One of the takeaway things that I want you to get from this animation is that what happens in the Arctic or in the Atlantic or any other ocean basin is not limited to that ocean basin. So what happens in the Arctic, these waters formed up kind of in the Arctic, and they're deep waters, and they're traveling all the way south along South America. They're coming around the coast of Antarctica. They're actually going to be brought up to the surface in the Indian Ocean. This takes a lot of time. Something like, to make it full circuit is about 1,500 to 2,000 years. But it does happen. And you can see these different layers form all throughout the ocean. They have different chemical properties. So again, it's really important to understand what's happening in the Arctic, not just for the Arctic's sake, but for the entire planet's sake. Okay. So just to show you again, the red arrows here are the surface currents. They're hot. They come all the way up into the Arctic Ocean underneath the ice cover. They're cooled off, at which point they sink down and they flow back out of the Arctic and back around. This is actually this body of water that's been cooled. Used to be at the surface. It's called the North Atlantic Deepwater. So the Atlantic Ocean is warming, but it doesn't sink quite as much. It moves into the Arctic and heats up the ice underneath. So not only is the Atlantic water being cooled by the Arctic, but the Arctic system is actually being warmed by the Atlantic. So what happens to the surface waters in the Atlantic plays a critical role in what happens to the entire Arctic ecosystem. So with the Arctic getting warmer, the air above is warming it, the water below is warming it, and so it's actually melting the ice from both sides. We're starting to see evidence of that heat coming in, kind of like a waffle maker. You know, you're cooking it from both sides. All right? So is this having an effect that we can actually detect? Yes. This year, the 2015-2016 season, that was the lowest maximum ice coverage extent ever observed, especially from the satellite imagery. So this actually occurred on March 24th, about a week or so ago. That was the largest. So the maximum extent is during wintertime, how far does the ice cover the Arctic Ocean? And so that's the most coverage that you can expect the ice to get, and that coverage was lower than it's ever been. And so what you can see here is that this gray line here is the average. We're going from month, January, February, March, April, May. This is during like the winter months. This is the extent in millions of square kilometers. You can see the average between 1981 and 2010 is this black line here. You've got the gray bars, which is kind of like the standard deviation. In 2012, this is the green dash line, kind of showing you what the extent of coverage was. Blue right here is 2016. This is just what we're coming out of. And this is today's data. Just pulled it off the website today. So you can see here that the blue line is well below the average. It's well below what it was just a few years ago in 2012. So we can actually see that ice shrinking. Okay. If you want to see how this kind of is impacted, this is actually data from this last cruise, courtesy of Jim Swift at the Scripps Institute of Oceanography. And so what you've got here is this is the mean temperature of the water that spills into the Arctic from the Pacific side. So you've got the Pacific, it's very narrow, so you've got a little bit of water that spills over the side and makes its way into the Arctic Ocean. In 1994, when they were out there, they measured the temperature of this water all the way from about 75 north, almost all the way up to the North Pole. And that's what this purple line is right here. So these are the temperatures, about 0.1, 0.2 down to 0. That's the temperature of that water mass. In 2015, all the way from the Pacific side, all the way up to the North Pole, it's 0.1 to 0.2 degrees warmer, and that does make a big difference. That's going to, not only is that going to bring energy, heat to the Arctic system, it's actually going to take that Pacific water, because it's a little bit warmer, it's going to be a little less dense. It's going to sit closer to the surface, which is where the ice is. That's what we want to not melt. So water from the Pacific is actually coming into the Arctic, and that water is also warming. All right. So the Arctic is changing. We don't even have a good snapshot of what it looks like now. So a baseline map is urgently needed, thus the drive to get to the Arctic as quickly as possible, team up with as many nations as we can, and try to survey the whole thing before things continue to change. We need a baseline to work from. These temperatures continue to change. The salinity of the waters in the Arctic are going to continue to change. We kind of need to know, we really need to know, what the chemical composition is of the Arctic Ocean. We need a map. So, one of the things that I'm particularly interested in is how these trace metals affect the biology. So I've worked diligently to try to partner up, as well as everyone on the GeoTraces team, to involve biologists who can bring in some extra information and some studies to complement our chemical data. So what we're looking at here is just a little cartoon of what what a typical piece of ice looks like. So you've got snow cover on top, you've got a lot of sea ice that sits down into the ocean, and then at the bottom of this, at the bottom of the ice, you actually have some algae growing, and they can get the sunlight through the ice, and they get the nutrients from the seawater below. So again, the health of the algae and the other things that grow they're at the base of the food chain, everything that grows up from them depends on the amount of ice coverage, because that's going to depend on how much sunlight comes through, but also their accessibility to nutrients. And what we've got here is like a little magnification thing. all these inside of the ice. It's not like, just like a solid block of ice. What happens when ice freezes is that the water will freeze, but it'll kick out the salt. And that's a process called brine rejection. So it'll keep kicking out the salt, and so the water that doesn't freeze just keeps getting saltier and saltier and saltier. Not only saltier, but also colder, because this water hasn't frozen and the other water around it just keeps freezing. So what happens when you get very cold water and very salty water? It actually gets very heavy. But these little channels are where all these little critters live. You've got bacteria, you've got these single-celled algae, and these diatoms, like the original picture I showed you, the nice little critter that has chloroplasts in there for photosynthesis. These are at the base of the food chain, and so other things are going to eat them, and other things are going to eat those. These all live inside the ice. So one of the things that ends up happening is that if that ice gets a little bit warmer, it may not melt the ice. The ice doesn't have to go away. But if it's warm enough to release all this trapped brine, that stuff, again, is really heavy. It's really cold. It's really salty. And as soon as those little pores open up, that brine just sinks right out and goes straight to the bottom. These guys don't have a chance to resettle. All right? So that's another concern. We actually saw this at a couple of the ice stations. We expected to find a lot of microorganisms living in the ice, and when they pulled the ice out, they found that it was just mostly fresh water, and all the brine that was inside had kind of dumped out, kind of just like a dead coral reef, just like a skeleton of what used to be. In addition to the things that live inside the ice, this is actually looking from under the ice back up to the underside. This is something called Melisira. It looks kind of like tendrils of kelp, maybe like a seaweed. This is actually just huge clumps and chains of those single-celled diatoms. So this is like huge. I mean, it can be like meters long. But it's actually made of little single-celled things that all live together. These kind of root like trees do into the ground, except they root to the bottom of the ice. Again, if the ice loses its grip, it loses some of its traction, these things, which are huge clumps, I mean, they don't even need the salinity, the heavy water, to kind of help them sink. They're so big and heavy that they just go straight to the bottom whenever they release. And so there were places where we went and we had like a little GoPro camera. You stick it down through a hole in the ice, look around. You don't see any of this stuff. But fortunately, at other stations, we did actually find quite a bit of this. And so we can see that it's there. This is a really important, base of the food chain. Provides a lot of food for everything else. And it's a pretty unique thing that grows on the bottom of the ice. So up on top, just snow, white, a few polar bears, some seals, but underneath there's all this activity going on. All right, so our goals for this trip, my goals as well as others, were to understand where the nutrients were coming from, how the ice is changing, and the effects on the phytoplankton. Those are kind of like my narrow little focus. So just want to show you, going up to the North Pole, this is what it looks like out on the ice. You have these pockets. This is where you don't have nearly complete coverage, but you have these pockets of molten snow and ice right on the surface. This is totally normal. It's fine. And you'll see these little tracks that you cut through here. This ice actually isn't too bad to cut through. That's loose. It's kind of broken up. It's got some little channels that you can find. It's not too bad. Now, when you get into the thick stuff, it's a little bit, it's a little different story. So I'm going to show you a video here. I apologize for the noise, but that's the point. This is what it's like to be on an icebreaker cutting through ice that's 1 meter to 2 meters thick. You ready? Oh, come on, back up. You like that rattling? Oh, anytime. It's a ship shaking.
Really? Wow. More cow?
All right. I think that's probably pretty good.
You gotta stop it.
Oh, I do. 2.5 months of that. I was so ready to sleep when I got off that ship. But seriously, that's what it's like all the time. Once in the open ocean, it's not too bad. You get through pretty quick. Even when the ice is loose, it's pleasant. You're like, oh, that's an interesting thing that's happening. But then when you actually get into the ice, all the time. You just got these incredible shutters. I mean, that's a big ship, and it is a beast. I mean, it will push some ice out of the way, and you could just feel the thing every step, every day, nonstop, this shaking just going through, knocking the fillings out of your teeth. But yeah, that's what it's like. So I'll tell you a little more in a second, but Neil actually did all of his science. He actually made analysis in the middle of that racket, so that guy deserves a medal. But you'll see, I mean, these are the conditions that we're working in. We've got to cut through the ice to get to the interesting parts of the Arctic. And again, why it's such a big deal that the Healy was able to make it unassisted. We didn't have to ask anyone for help, which was nice. So once we actually had some ice stations that got into the ice, what we would do was send teams out. They would scope, the Coast Guard would scope out the area. They'd test to make sure the ice was sound enough for us to walk on. And then we'd send out teams, and each team kind of had a little section that they worked in. So we've got two individuals here. This is Carl Lamberg, now at UC Santa Cruz. This is Kyle Dillaplane, who's actually a graduate student at the University of Alaska Fairbanks who specializes in sea ice biology. He has all the great tools. He has some cool stuff. And so he's got this monster of a drill here. And Kyle's, I don't know, what do you think, Neil, about 6.5 foot? He's probably on level with Bill. So Kyle's, giant, tall guy with this huge thing because he's got to cut through about 2 meters of ice. So he's up there with this drill just plowing right through, coring out. And so he and Carl work together for most of the cruise. You could see here, once they collect the core, they pull it out intact. And you'll see that Kyle is not wearing a glove. That guy spends a lot of time out there on the ice. He's kind of used to it. He never got frostbite as far as I know. He was good. Never lost a finger or anything. But he's just, in order to get all the ice out and to keep it intact, he'll, once they core out this hole, which is probably about 4 inches across, and he needs to make sure he gets every little bit of ice out, he'll just stick his hand right down in there and grab it out. Not me. So these guys are collecting parameters that, collecting samples for parameters that are not contamination prone. So he can use this kind of super metal core. They can wear their, what I would call street clothes out in the Arctic. This is their foul weather gear. Their super suits keep you nice and warm. Kyle can grab the ice with his bare hands and it won't affect anything. If you want to do the trace metal stuff, you got to wear all this gear plus a Tyvek bunny suit. So, they didn't make any friends with polar bears, so, but they did. It was interesting to be out there and say, hey, where's Anna and Rob? And you're like, oh, there they are. I can barely see them, only the tops of their heads. So you can see that their sampling system here actually is built of titanium, which doesn't shed. It's metal, but it's one that it doesn't shed off into our samples. And the sleeves are actually made out of a Teflon, so that they don't have, Teflon's one of the best substances we use for collecting trace metal samples because they don't have any metals in them. You can really aggressively clean them with some strong acids, and that really helps pull out all the dirt that's embedded in the walls. And so these guys would stay dressed like this for like 8 to 10 hours. People would run coffee to them every now and then. But these guys, once they were out there, they were committed. And Anna and Rob are actually both at University of Alaska Fairbanks as well. So you can see everybody kind of has their own specialty, and we all work together here in the U.S. and also internationally. So, yeah. So this, when we want to collect water samples, we actually have a different system. So the ship, you saw it as we were cutting through the ice, it broke up a bunch of pieces, and then would back up and then dig out some of the, you saw it was more open behind it. So when we were ready to collect samples that were not at the ice level, but way under the ice and down to the seafloor, we would use this kind of rosette system. And I will press play. There's no sound here, don't worry. Once was enough. Hold on. Let's see if I can get it to work. Oh, here we go. Great. So this is actually another one of those GoPro cameras put off the side of the ship. And there's the big piece of ice stuck in the props there. And so we'd actually deploy this equipment off the back. I want you to see it. So you can see the ice surrounding us. There's a team of no less than 4 or 5 Coast Guard personnel outside at all times with long kind of spears to kind of poke away the ice, keep it from getting caught in the instrumentation. You don't want to make it all the way to the North Pole and then bump this into something, drop it to the bottom. And so each one of these bottles will close at a different depth. And so we send it all the way to the bottom, trip the first one, move it up a little bit, trip the second one, move it up a little more, trip the third one. We bring them all up. And I'll show you the space that we actually collect the water from. Keep it nice and clean. All right. All right. So the Canadians who are out there had a unique set of problems, these poor guys. So this is the sampling system. They've already got theirs in the water. And I have muted the sound. You can look it up later if you like. But let's see if I can get this to play here. There we go. So yeah, they're cute, right? So that's 1,500 pounds of toddler right there. Those polar bears are super curious. I'm sure they're very nice, well-intentioned animals. But just like a toddler, they explore by putting everything in their mouth. And so there's a lot of stories I could tell you about polar bears. We didn't have any real encounters with them when we were out there. A couple of them came up pretty close to the ship, and we did have to move once or twice. But fortunately, nothing happened when we were out on the ice. They're Coast Guard personnel who were very well trained in what they do, who keep an eye out at multiple levels on the ship, also out on the ice with us, so we're very well protected. Because these guys don't joke around. They're about 1,500 to 2,000 pounds. And again, the way they kind of explore their environment is to put it in their mouth. So thankfully, this line right here is Kevlar. So it's a Kevlar line, same stuff you would make, you find like bulletproof vests. So it's very strong stuff, but there's some sensitive electronics that run through the middle. And so the Kevlar supports it, but the electronics in the middle allow us to communicate between the deck and the sampling system. So these guys get in there and start to chew on it. Even if they don't cut the line, they could mess up the electronics in the middle, and that'll ruin your day. So once we do successfully recover the bottles, we bring them back onto the ship, and we've got them lined up everywhere. So this is me in what is a laboratory van. So it's like a shipping container that's outfitted on the inside as like a laboratory. So these things are nice and portable. We ship them all over the planet. They get craned onto a ship, bolted down, and inside it has kind of like what the MagLab has in the geochemistry, that HEPA filtered air, ultra high purity air. I am wearing gloves, but I'm actually a little more relaxed in here. We do keep, there's only like there's no pass through here. There's only like 2 or 3 people are allowed in the van at a time. So this is how we collect our super clean samples. And when we're at sea, we have these portable labs. I'll show you Neil's setup in just a minute. But you can see here, we have these little filters that come off of these spigots at the bottom. So we overpressure the bottles. We give them some pressurized air, not so they spray out. We want to be gentle because we don't want to disrupt the biology that's in there. Put too much pressure, they'll pop open. And so we very gently push them out. I'm actually collecting samples for the mercury team. So these guys did a lot of different mercury analysis when we were up there. So this is Neil and his, not everybody had the luxury of having like a sampling van. So what we also do is we get those plastic sheets that you would put down on the ground like when you're painting. They turn out to be really particle free, they're great. And so we would build these, we call them a bubble. And so you have this plastic, and you basically build a little room out of it. You get a bunch of those HEPA filter fans and line them up at the top, and you blow air in, so it's nice, clean air blowing in. And sure enough, if you make sure all the seals are pretty good, you only have a door that lets you in and out, that thing will fill up just like a bubble. So that's what Neil's got going on here. He's actually taken a step further and taken another HEPA filter to blow over his samples, because once we collect the samples in the van, we'll bring them over to Neil, and then he'll open them in this very clean workspace, and he'll make these shipboard zinc measurements. And so the trip, again, we just got back mid-October. So that's a couple months ago, but it takes some time to get everything shipped back. So I don't have a lot of data to share with you, but I can at least show you what Neil was able to accomplish on the ship. So he measured dissolved zinc. Remember, that was one of the important ones. It helps in carbon cycling. It's very contamination prone because the ship has the sacrificial anodes that are just kind of leaching zinc the whole time. It is a trace nutrient, again, runs carbon enzymes. It's also a tracer. It turns out that the Pacific waters have a different zinc signature than the waters in the Arctic. You can actually follow where the Pacific water is coming in by looking at the zinc signal. And also, not only is zinc a trace nutrient, but it almost has like a big brother. It acts a lot like silicon, which is a major nutrient. And silicon's important because that's what the diatoms build their little glass shells out of. And so those things that I was showing you before, those nice little pictures of diatoms, that's a kind of glass right there. We actually call it biogenic silica. It's just like quartz. And so zinc acts like silica. Silica is used to build those shells. So they have a unique relationship in the ocean. So what I'm going to show you is, again, here's the map. And so what we did, again, was we went up to the North Pole and came back. So you'll see the next chart, the next plots I'll show you are shown left to right. But you'll see it going from the start of the trip to the North Pole and then back south, but it'll be in one straight flat panel. All right, so what we've got here is, if you're coming from the Pacific side, this is the Chukchi Sheet, a nice narrow shelf. It's like 25 to 50 meters between the Pacific and the Arctic. Again, this is, you know, where when it gets nice and dry, that's where people can walk straight across the Bering Strait. So it's relatively shallow. And then what you can see here is the dissolved zinc signal. You have this huge plume of dissolved zinc that comes right off the shelf and makes it, how far? This is 90 north. This is probably about 80 to 85 north. It actually makes it really far north. I mean, that's a huge signal. And these concentrations are not what you would expect to encounter in the surface. Usually you would expect to find something about 100 times lower than that. And this is the concentration you would expect to find kind of close to the bottom after all the plants who take up the zinc die, sink to the bottom, and then release all their nutrients back into the water. And you see the same thing heading to the North Pole and also coming back. So this is a different part of the shelf, a little bit further east, closer to the Canadian archipelago. And again, you see that same signal of zinc coming off the shelf. And if you look, just like you would expect, we knew that there was this relationship between zinc and silicate. And actually, this is the silicon concentrations right here, actually matches very well. So you see the zinc is high at this point. Silicon matches very well. Same thing coming back into the shelf on the return trip. So this is just some of the, I mean, this is the freshest data you could possibly have. So Neil actually gave a great talk on it about a month ago. First time this data had been shown ever. I mean, this is really cutting edge stuff that's going on at the MagLab that we were able to be a part of. So the type of work that I do, I care not about what stays dissolved in the water, but how much ends up in the particles, i.e. where the phytoplankton live. And so Neil will look at it from one side, what's left over in the water. I'll look at it from the other side, what makes it into the plants. And so we'll see if we'll try to build a budget, a balanced budget of how these things move. So this is a technician from Bigelow Labs in Maine. This is one of the persons on our team. So what she's got here is the coolest microscope I think I've ever seen. This thing is amazing and takes the best images, really great pictures. All the pictures I've shown of phytoplankton were done on this trip with this microscope system. And it'll do a lot more. I can't wait to show you. So what you've got here is Sarah. She'll go through and filter out a bunch of these phytoplankton or microbes onto a slide, a specialized kind of slide. And then she'll look to see, she'll find some interesting characters in there. And she'll take pictures of them. And the slide will actually remember where these things are. So what she'll do is then take them. This is, again, looked up at that thing very, at the phytoplankton. The diatoms very well magnified. Again, you can see the kind of the glass shell here. And the little green bits are little tiny single cell things that have a lot of green chloroplasts. So one of the concerns is this stuff, all this biology we see in the ocean, is it alive? Is it dead? What are we looking at here? Because again, with that signal that we see coming off the shelf with the zinc into the ocean, it takes a lot of material that's at the bottom and stirs it up and then kicks that out. It's almost like smoke in the ocean. So the way you tell if this stuff is alive or dead, you look for the green, but you can also shine fluorescent light on it, and you'll see the stuff that's alive glows red. The stuff that's dead, i.e. didn't have any chlorophyll in it, doesn't show up at all. So the next thing that we do, and we don't have any of this data yet, but we're getting to that. This is where the project's going. So this is the fresh stuff that we just got from the Arctic. On the North Atlantic cruise, did the same thing. So what you've got here are a bunch of different microscope pictures, like just visual pictures of these phytoplankton. They've already been tested for their fluorescence to see if they have chlorophyll or not, whether they're alive or dead. So the guy that I partnered with up at Bigelow, his name is Dr. Ben Twining. So yeah, Ben Twining works at Bigelow. I love it. So what you see here is he has a very specialized technique where he can actually measure not just the total concentration of metals or elements in the microbes, he can actually target at such fine resolution where the metals are in a cell. And this is what the power is. So if you look at this cell, here's phosphorus, here's sulfur, and here's iron. They all kind of look the same. They all kind of glow right here in the middle with this red spot, and they're cool around the edges. If you look at this guy here, here's the phosphorus. It's all localized. It's a really long, thin kind of, what is it? Diatom. It's very thin, and so the phosphorus is located right here. But if you look at the sulfur, it's kind of spread all along the length of the phytoplankton, and then the iron also appears to be spread out. And you know I'm building up to it. So you see this one here, you can see a lot more differentiation throughout the cell. So you've got a hot spot of phosphorus here, a cool spot, and then a little more phosphorus down here. And then look at the sulfur, just spotty all over. And then if you look at iron, none of that overlaps with the sulfur or the phosphorus. What this tells us, not only does it give us a nice way to kind of balance how much phosphorus there is, how much iron there is, and put together ratios of like how much iron to phosphorus, how much iron to sulfur. These are important tools that we can use to kind of tease out the health of the phytoplankton. But also it tells us if these things overlap, if they're associated with each other. So there's some special enzymes that use iron and sulfur together, chemically bonded, to run certain mechanisms. This one has a ton of sulfur, but not a lot of iron in it. You know, what is it that's making, what enzymes are here versus what's here? And so you can see that there's a lot of information to be gained from this. Also looking at trace minerals, not just what's dissolved left over in the water, but what the phytoplankton take up and where they're using it. So I'm going to wrap up right now with what I think is one of the biggest challenges, something we heard a lot about in the news when we were there, is that one of the big challenges to the U.S. research in the Arctic is that we're kind of limited on icebreakers. I think you can see how critical it is that we have a good fleet of ice-breaking research vessels that we can take into these polar regions. So this was a newspaper article that came out September 7th, 2015. I thought it amazingly coincidental. That's the same day we hit the North Pole. So if you look here, I'm going to pull out some of the information. The U.S. only has 1 to 2 operational icebreakers. So if you want more info, you can go to iceflow.net. The Healy is only considered a polar capable vessel, which means that it's good during the seasons that are most favorable for the lowest ice. The Polar Star is a polar class vessel that can go year round, but it's in dry dock, or not dry dock, but it's in dock right now getting some repairs. Unfortunately, the Polar Sea has been broken since 2010. Looks like it's going to be too expensive to repair for a while. So we have 3 vessels, 2 of them are working. The one that we took was polar capable. The only other one we have is polar class. To compare that, the Russians have 40 active icebreakers. 27 of them are open ocean going. 10 of them are nuclear powered. And they have 11 that are planned and under construction right now. So I think without getting into it, because I'm not sensitive to these things, this has much bigger implications than just science, economic, military. This is a big deal that we put some resources back into Arctic research. So again, just showing you this was one point to consider is that the Canadians participated, the US team, the Germans, and the Dutch, but you'll notice there's this huge area right here, which is where 8 of the 10 biggest rivers dump into the Arctic, and they're all in Russian territory. We really need to figure out a way to get some cooperation going there. With that, I want to thank you for your patience and your time. I hope you enjoyed the talk. I certainly did. Be happy to take any questions. Yes, ma'am.
Actually, I have 2 questions. The first is, when you get all of your data eventually, what do you hope to be able to tell from it? And the second question is, given concerns about the Arctic ice disappearing and thinning and destabilizing, all of these icebreakers, are they contributing? Do you have any concerns about them contributing to the destabilization of the ice shelf?
Absolutely. So we had 2 great questions up here. Number 1, what do we hope to do with all the data? So it just happens that Friday was a major deadline for GeoTraces. It's the second time that we've had a major, a submission deadline just for the data. Not the publications, but for the data. And that's called, there's something called the intermediate data product. And so there's a 2014 version and the 2017 version. The data was just, the deadline was just this past Friday. A lot of people very busy. And so what they do is they take all their data, every bit of concentration data that they've generated, and they send it to a central database. And so this data, these people that run this database, they'll mine through the data, make sure everything is collected to the appropriate quality, all the analyses were done correctly, and then what happens is they'll make that publicly available. What you can do with that data, like what Neil had done, I didn't want to steal his thunder, and he's the expert on it, but what Neil has done, I told you a little bit about the zinc and the silicate relationship. Well, it turns out there's subtle differences depending on whether you're in the Pacific, the Atlantic, the Arctic, and things will modify. Things will change that ratio slightly if they move up against the surface, like if they move against the shelf, a coastal margin. You know, there's different things that can modify that signature. So what Neil does is he just takes these huge databases of the zinc and the silicate data, and he combines the two, and he looks for little tiny differences. That would not have been possible without the extensive coverage that we see from all these international participation. So the second question you had was, what kind of concerns are there about so many icebreakers coming through the Arctic, especially when the ice is getting a little less stable, a little weaker? And so one of the things that I don't know, I haven't heard any real concerns about it, you know, it contributing to breaking up the ice. That ice is pretty tough, and we're a little tiny ship still in a big ocean. With increased traffic, though, that could be a concern, especially with pollutants, because that's still a pretty pristine area for the most part. There is a lot of industrial stuff that makes it from North America and Northern Europe that can get carried up into the Arctic. I mean, you can see some of the videos, even with our ship smoke, you can see like this little yellow haze and kind of like a bullseye off on the horizon. I mean, we've got to power it to get it up there. And so you can still see like some of our own ship smoke coming out. So I imagine with increased traffic will become some increased pollution through those areas. All right. Is there another question? Yes, Beth has a question.
So that warming effect that you mentioned with the warmer water melting next to below and the warmer air melting next to above. We had a warmer El Nino or more aggressive El Nino this year. So is there any like quantification as to how much that effect is compounded by the strong El Nino effect, or is it overarchingly from the ocean?
You know, I was trying to cram for my talk tonight and read up on all this stuff. I am probably not the best person to answer that question. So best question was, if there's a strong El Nino, how does that affect some of the air? So the thing like El Nino that's up in the Arctic is called the North Atlantic Oscillation. I think you hear it on the Weather Channel, something like the polar vortex. That's also a big deal, right? We've been in a positive phase for a long time that has some sort of effect on it. Again, I tried to read as much as I could for tonight, anticipating that Beth, she is not a plant, but I was afraid someone would ask that question. It is a lot more, I'm not quite as sensitive to all the details, but what I did read is that the El Nino and North Atlantic Oscillation and also the monsoons in Indian Ocean are very closely related. But I think the guys in meteorology would probably be better to ask. But yeah, I know they're all related. You're very welcome, Beth.
Any other questions for Pete? Okay, let's have one more round of applause.
Thank you.